Hello everyone, in this video we will talk about the pathway of heme synthesis and all the disorders associated with it, which are known as porphyrias. The organs involved in the synthesis of heme are liver and in the dividing cells of the bone marrow. Keep in mind, heme synthesis is absent in mature erythrocytes. If we talk about the organelle in which heme synthesis happens, it is both mitochondria and cytosol. So heme synthesis is one of the processes which involve both the mitochondria as well as the cytosol. Now let us look at the reactions involved. In the first step, succinyl coenzyme A and the amino acid glycine both combine to form aminolevulinic acid. This reaction involves the enzyme delta ALA synthase or delta aminolevulinic acid synthase. This enzyme requires pyridoxal phosphate or vitamin B6 as a cofactor. So a deficiency of vitamin B6 can lead to anemia. The second step is formation of porphobilinogen from aminolevulinic acid. So two molecules of aminolevulinic acid combine in the presence of the enzyme ALA dehydratase and give rise to porphobilinogen. During this process, two molecules of water are released. The next step involves the formation of the porphyrin ring. So first, 4 molecules of porphobilinogen will combine in the presence of the enzyme porphobilinogen deaminase to give hydroxymethylbilane. This hydroxymethylbilane in the presence of the enzyme uroporphyrinogen 1 synthase forms uroporphyrinogen 1, leading to the formation of the porphyrin ring. Then uroporphyrinogen 1 is converted to uroporphyrinogen 3 by the enzyme uroporphyrinogen 3 co-synthase. Uroporphyrinogen 3 becomes coproporphyrinogen 3 with the enzyme uroporphyrinogen decarboxylase. Coproporphyrinogen 3 becomes protoporphyrinogen 9 in the presence of the enzyme coproporphyrinogen oxidase. Now protoporphyrinogen 9 will become protoporphyrin 9 in the presence of the enzyme protoporphyrinogen oxidase, and in the final step protoporphyrin 9 is converted to heme within the presence of the enzyme ferrochelatase, and in this step iron is added to the porphyrin ring. After looking at the chain of reactions involved in the synthesis of heme, now let us look at the porphyrias that arise due to the deficiency of the particular enzymes. All of the porphyrias can be divided into two types based on the manifestations that they exhibit. So they could have either neurovisceral manifestations or manifestations due to photosensitivity. In neurovisceral manifestations, we can have complaints like psychiatric symptoms, abdominal pain, hypertension, tachycardia, and constipation. Whereas in photosensitive symptoms, we can have skin lesions such as bullae, erythema, and erosions, which appear when the person is exposed to light, especially sunlight. So deficiency of the enzyme delta ALA synthase leads to X-linked erythropoietic protoporphyria. Deficiency of ALA dehydratase leads to ALA dehydratase deficient porphyria. Now deficiency of ALA dehydratase can also be caused by lead poisoning. And the main manifestations involved here are neurovisceral, where the patient will have abdominal pain, tachycardia, hypertension, and constipation along with psychiatric symptoms. You can remember it this way. The word lead has L and D in it. So ALA dehydratase enzyme is what is affected in lead poisoning. The next condition is acute intermittent porphyria, which is caused due to a deficiency in the enzyme porphobilinogen deaminase, which also involves abdominal pain, vomiting, CVS features like tachycardia and hypertension, and neuropsychiatric symptoms. In congenital erythropoietic porphyria, there is a deficiency of the enzyme uroporphyrinogen 1 synthase. And in this condition, now we start to see photosensitivity symptoms. One point to remember here is that all the porphyrias are autosomal dominant in inheritance, but congenital erythropoietic porphyria is autosomal recessive. The next enzyme is uroporphyrinogen decarboxylase, which is deficient in the condition Porphyria cutanea tarda, which also involves skin features and photosensitivity, causing bullae, erythema, and erosions. The next condition is hereditary coproporphyria, in which there is a deficiency of the enzyme coproporphyrinogen oxidase. This is obviously easy to remember because the condition itself has the word coproporphyria in it, and the enzyme also has coproporphyrinogen in it. The next condition is variegate porphyria, in which you have both neuropsychiatric as well as photosensitivity symptoms. And this is caused by the enzyme protoporphyrinogen oxidase. Finally, the deficiency of the enzyme ferrochelatase is seen in the condition erythropoietic protoporphyria, where there is only skin manifestations, and lead poisoning can also cause a deficiency of this enzyme. Now, if we look at the regulation of this process of heme synthesis, we note that the process happens in two places, the liver and the RBCs. In the liver, heme formation is required to form all the cytochrome enzymes, whereas in the red blood cells, heme formation is obviously required for the formation of hemoglobin. In the liver, inhibition of heme formation is done by heme itself. And this is done by the processes of feedback inhibition, repression, or by inhibiting transport. So heme will either inhibit the formation of further heme in the liver by feedback inhibition or by repression of the genes which are involved in the synthesis of the enzymes which are required for heme synthesis, or by inhibiting the transport of the metabolites required for the synthesis of heme. And substances which promote heme synthesis are drugs like phenobarbital, insecticides, and carcinogens. In the RBC, the presence of heme increases the formation of globin so that both of them can combine to form hemoglobin.